Hello everyone, welcome back. Today I'm here with this 14th part of this questionnaire and I will discuss some questions from biochemistry in this part. Let's get started. The first question is net gain of ATP molecules in complete aerobic breakdown of 1 molecule of glucose is. Our options are 32, 34, 36 or 40. So it's right answer is 36. I will discuss this in detail and this will come under the topic that is called respiratory balance sheet. Respiratory balance sheet. So there is production of ATP at the substrate level and other production of ATP is known as oxidative phosphorylation. So during substrate level phosphorylation that we can see in this flow chart. So the first step in respiration is known as glycolysis. It is common for both aerobic and anaerobic respiration. Glycolysis में जो ग्लूकोज है वो ब्रेक डाउन होता है दो पायरुवेट में, and during this process 2 ATPs are used or utilized. 2 ATP यूज होते हैं और 4 ATP प्रोड्यूस होते हैं. So we can say here the net gain is 2 ATP. And in addition to ATP, 2 NADH are produced, and normally a single NADH molecule when enter in the electron transport system, it produces 3 ATP, while this FADH2 produces 2 ATP. But as in case of eukaryotes, जो NADH हैं they are produced in the cytoplasm, cytosol में produce होते हैं, so they have to enter inside the mitochondria. तो ये जो NADH है इन्हें enter करना पड़ता है माइटोकॉन्ड्रिया के अंदर. For this reason 2 ATPs are utilized. So normally 2 NADH should produce 6 ATPs. Normally 6 ATP produce होने चाहिए, but 2 ATP utilize हो जाते हैं इसके ट्रांसफर के लिए inside the mitochondria, so there is net gain of 4 ATP. While there is second step in in this respiration that is called, in aerobic respiration, that is called link reaction, or it is also known as oxidative decarboxylation of pyruvate. So it involves removal of carbon dioxide to produce this acetyl coenzyme, and during this process again 2 NADH produces, and they will produce finally 6 ATPs in electron transport chain. Then the third step in aerobic respiration is Krebs cycle. During Krebs cycle there is formation of 2 ATPs at substrate level, that is called substrate level phosphorylation, and these 2 ATPs can be directly utilized. Then there is production of 6 NADH molecules, NADH2 molecule, they will produce 18 ATP, and 2 FADH2 they will produce 4 ATPs. So if we count them, so there is total production of 36 ATP. Normally there is 36 to 38 ATPs which are produced during aerobic respiration, as in case of eukaryotes as well as prokaryotes, but the formation of NADH during glycolysis will decide while it is 36 ATP or it is 38 ATP. In most of the eukaryotes, जो NADH है that is produced in cytoplasm and then it is transferred to the mitochondria, so that is energy consuming process, और उसमें 2 ATP utilize होते हैं, so the net gain is 36 ATP. So 2 ATPs are produced by substrate level phosphorylation and in glycolysis, and 2 ATP are produced at substrate level phosphorylation by this Krebs cycle, and finally 34 ATPs are produced during ETS, the electron transport system. So there is total net gain of 36 ATPs. So next question is, protein part of an enzyme is known as. Our options are holoenzyme, prosthetic group, apoenzyme or cofactor. So enzyme is formed of 2 parts, that is one is proteinaceous part and second one is the metallic part or non-protein part. So this protein part is known as apoenzyme. इसे कहा जाता है apoenzyme, और ये simply एपो एंजाइम एक्टिव नहीं होता है, इसको फंक्शन करने के लिए इट रिक्वायर दिस कोफैक्टर दैट इज़ नॉन प्रोटीनेशियस पार्ट और एक्टिवेटर विच मे बी मेटालिक आयन और एनी अदर ग्रुप. तो ये जब इस एपो एंजाइम पे जुड़ता है then it will form this enzyme which is known as holo enzyme, and this is the active enzyme that can reacts with substrate and produces the product. So here I will discuss about the classification of enzymes according to its function. So first type of enzymes are oxidoreductases. So these enzymes helps in transfer of hydrogen, catalyzes oxidation reduction reaction as conversion of lactate to pyruvate by lactate dehydrogenase. So this is the enzyme. So it is oxidoreductase enzyme that helps in redox reactions, carrying redox reaction, that is oxidation reduction reactions. Second enzyme class of enzyme is transferase. So it catalyzes transfer of some groups, that is carbon, nitrogen or phosphorus containing groups, such as conversion of serine to glycine by serine hydroxymethyl transferase. Then third category of enzymes on the basis of its function is hydrolases. As the name indicates, they helps in breakdown reactions, so they catalyze cleavage of bonds by addition of water. तो वाटर की इन्वॉल्वमेंट से ये ब्रेक करते हैं बॉन्ड को. फॉर एग्जांपल वी हैव यूरिएज एंजाइम and it converts urea into CO2 and NH3. This is very important enzyme जो कि यूरिया को convert करता है कार्बन डाइऑक्साइड और अमोनिया में, and this enzyme require one cofactor that is called nickel. That is very important. So nickel की presence में ही ये enzyme काम करता है. The next fourth class of this enzyme on the basis of this function is lyases. Again ये भी break down करते हैं but ये water को involve नहीं करते, so cleave without adding water, catalyze cleavage of carbon-carbon bond or carbon-sulfur bond and certain carbon-nitrogen bonds. Such cleavage of pyruvate, such as cleavage of pyruvate by pyruvate decarboxylase into acetaldehyde. So ये जो reaction है जिसमें pyruvate को convert किया जाता है acetaldehyde में, so that reaction is carried out by the enzyme which is commonly known as lyases. The next class of enzyme is isomerases. They helps in isomerization reactions, so they catalyze racemization of optical or geometric isomers, such as conversion of methyl malonyl coenzyme to succinic coenzyme by methyl malonyl coenzyme mutase. So these enzymes helps in isomerization reaction. The next category of enzymes on the basis of its function is ligases. So these are commonly known as molecular glues. So ये glue की तरह काम करते हैं, means they helps in joining the bonds. So ATP dependent condensation of two molecules, it require energy. For example, acetyl coenzyme carboxylase. So it catalyzes formation of bond between carbon and oxygen, sulfur, nitrogen, coupled to hydrolysis of high energy phosphatases, such as conversion of pyruvate to oxaloacetate by pyruvate carboxylase. So it helps in addition, or it will helps in formation of bond, and this reaction is ATP dependent. Next we have some specific sites on this enzyme that is called active sites, or inactive sites पे substrate bind करते हैं, and then they will form enzyme substrate complex, and finally enzyme gets separated out and product is formed. So these active sites are the site where the substrates will bind, a particular substrates will bind, and enzymes are highly specific in nature. जो enzymes हैं और biocatalysts हैं, they are highly specific. Then one important term which is related to enzyme is known as the turnover number of an enzyme. So turnover number of enzyme simply एक ऐसा नंबर जो जिसमें एंजाइम कन्वर्ट करता है सबस्ट्रेट को प्रोडक्ट में इन अ गिवन टाइम. सो टर्नओवर नंबर आल्सो टर्म्ड एज KCAD. Catalysis is defined as maximum number of chemical conversions of substrate molecules per second that a single catalytic site will execute for a given enzyme concentration. So enzyme कितने टाइम पे कितने substrates को प्रोडक्ट में कन्वर्ट करता है, that is its turnover number. So KCAT is equal to Vmax by ET. For example, the fastest enzyme is carbonic anhydrase, और इसका जो turnover number है that is 4 lakh to 6 lakh per second, which means that each carbonic anhydrase enzyme molecule can produces up to 6 lakhs molecules of product bicarbonate ions per second. The next question is, enzyme acts by, our options are lowering the activation energy, increasing the activation energy, conserving the activation energy, or all of the above. So its right answer is lowering the activation energy. Basically, जब enzyme catalysis करते हैं, so this is a graph which shows this enzymatic reaction. And on the x-axis it shows reaction progress, and on the y-axis it shows energy. So basically जो ये barrier है, this barrier, this is the substrate and this is the product. So substrate को product में convert करने के लिए इस energy barrier को pass करना पड़ता है, and this energy barrier is known as activation energy, which is required to convert this substrate into product. And enzymes basically helps in reduction or reducing this activation energy and lowering this activation energy to speed up the chemical reaction, biochemical reaction. So this is how the enzyme works on substrate and converted into products. So enzymes basically are the biocatalyst and lowers the activation energy needed to start a chemical reaction, so they speed up the chemical reaction. It does not increase how much product it is made, only how quickly it is made. So enzyme and proteins that acts as biological catalyst. Basically जो enzymes हैं, these all enzymes are proteins with some exceptions, and they are commonly known as biocatalysts. So basically they works by lowering the activation energy of this biochemical reaction. So without enzyme, जो products हैं उन्हें इस energy barrier को pass करना पड़ता है and require a lot of energy and time, while when enzyme is used, it lowers this activation energy, decreases this activation energy, and speed up the biochemical reaction, and products are formed. The next question is, competitive inhibitor that stops the activity of an enzyme succinic dehydrogenase is, our options are malonic acid, succinic acid, fumaric acid, or oxaloacetic acid. So basically जो competitive inhibitors होते हैं, they are just similar to that of substrate, and they occupy all the active sites of enzyme. वो enzyme की जो active sites होती हैं उनको occupy करते हैं and blocks the site for substrates. So enzyme is unable to convert the substrate into products. So these inhibitors are known as competitive inhibitors, means they will mimic the substrate और ये occupy करते हैं active sites on enzyme. So its right answer is malonic acid. So malonic acid एक ऐसा substrate है जो succinic dehydrogenase enzyme के substrate की तरह है and it blocks its activity. So competitive inhibitor is one that competes with substrate molecule for the active site of the enzyme. तो compete करता है ये, and it resembles the natural substrate and combines reversibly with the free enzymes at the active site and blocks these active sites, जिसकी वजह से जो enzyme है that is unable to convert substrate into the products because substrate is unable to bind on the active sites of the enzyme. So malonate is an example, malonic acid. So this enzyme inhibits succinate dehydrogenase activity, and the structure of malonate is similar to succinate. Basically जो substrate है इस enzyme का, that is जो enzyme succinic dehydrogenase है, इसका substrate है succinic acid, but succinic acid की तरह ही जो दूसरा substrate that is called malonic acid, it acts as a competitive inhibitor और ये occupy करता है इस enzyme की active sites को and blocks the sites for the succinic acid. So this enzyme is unable to convert this succinic acid into product, and this is an example of competitive inhibition. So competitive inhibition में जो enzymes होते हैं उनकी जो active sites होती हैं, they are occupied by this inhibitor or competitive inhibitor, which is similar to that of substrate, and substrate is unable to bind on the active sites and enzyme is unable to convert this substrate into products. So that is called competitive inhibition. While in case of the non-competitive inhibition, so जो inhibitors होते हैं वो enzymes के किसी portion पे bind करते हैं and changes its geometry, और इस enzyme की shape को change करते हैं, और जो active sites होती है वहाँ पे substrate bind नहीं कर पाता है. So this is known as non-competitive inhibitors. So these non-competitive inhibitors changes shape of enzymes so that it cannot bind to the substrate. So जो substrate है वो इस पे bind नहीं कर पाता because there is geometrical change in the enzymes. So this is succinate, and normally this enzyme, that is succinate dehydrogenase, it acts on this substrate that is succinate and converts it into fumarate. But When this malonate is present, it can occupy the active sites of this enzyme and this reaction is unable to complete or succeed. So a cumulative inhibitor of the next question is Heme and globin part of this important pigment which is known as leghaemoglobin that is found in the root nodules, and it acts as oxygen scavengers and provide oxygen free environment for nitrogenase enzyme. So it is synthesized on our options are bacterial and host plant genome respectively. Bacterial genome, host plant genome, or host and bacterial genome respectively. Clearly I'm unable to find its answer, but the most probable answer is bacterial and host plant genome respectively, because that Heme part of this leghaemoglobin under normal physiological condition is synthesized by bacteroids, and bacteroids are the mature bacteria which invade the roots of legumes, and this globin part, which is protein part, this is synthesized by the ribosomes of infected cells. Then Heme and globin are assembled in the plant cell cytoplasm. So the most probable answer is bacterial and host plant genome respectively. So this was all about for our today's discussion about these questions. Hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, you can leave it in the comment section. Thanks for watching. Have a great day.